Good evening, everyone, and thank you for joining our Weizmann Annual Lecture 2020. This year, it's 20th anniversary of this symposium. 20 years ago, my predecessors had this idea, and it was a big success, but it's the first time that we do it only virtual. So for one, that has the benefit that a lot of people of our friends around the globe can participate. It has the disadvantage that we cannot offer a nice little dinner at the end, but I hope you have the fridge nearby and can serve yourself and get some nice food afterwards. I really want to thank you all for your loyal friendship with the Institute and the Swiss friends who support the Weizmann Institute. It's really great to have you with us. Just some technical matters. We have left really ample time for questions and answers at the end of our presentation today. So what you can do, and you can start then immediately, put questions into the chat room. The chat room, is at the bottom of the screen. You can click on the icon chat and put in questions. You can write it in English, in French, in German, and we'll then try at the end to walk through those questions and to answer them. So we really hope by doing this that we have a lively and exciting event today. Also we are not all together in the same room in the aula at the University of Zurich, but nevertheless, that really people can interact, and it's not just like being in the movie. So from that point in time, I just want to pass on the floor to His Excellency, the Ambassador of the State of Israel here in Switzerland, Mr. Jacob Kaidar. Jacob, please, the floor is yours.
Thank you very much, Eric. Well, dear Eric, dear Professor Selenko, dear Zohar, Hadar, and all our friends, ladies and gentlemen, this year is a bit strange year for us. I mean, I enjoyed every time very much the event of Weizmann Institute, to hear the lectures and to be physically, to meet the people, and of course the gala dinner afterwards. But this time it's a bit different with Corona times. But still, even on Zoom, it's very nice to see all of you, even if it's through the computer screen. And I wish you from here a lot of good health and be strong. This year, my daughter, Tal Keidar Haran, joined a laboratory in Weizmann Institute. It's called the Keren Laboratory for System Tissue Pathology. I have no idea what they're doing in Weizmann Institute, and it really fills me with a lot of pride, and it gives me a bit more connection to Weizmann Institute, even more than before. And we all know that Weizmann Institute stands at the higher end of the research in the world, and even in this very difficult times of Corona, it makes a lot of contribution to mankind in finding solutions for this illness and for many other issues. We try to help as an embassy for the connections, for the collaboration between the institutes, between researchers in Israel and Switzerland, and especially between Weizmann Institute and many institutes in Switzerland. And I assure you we will continue to do it. You know that unfortunately, this is maybe my last event with you, as I'm scheduled to leave Switzerland at the end of January. If you insist, I can ask for more time, but I don't think they will give me. So it really was a pleasure to be with you in every conference in Switzerland, also in Liechtenstein, by the way. And I learned to know you. It has been a great honor to be with all of you and to see all your assistants, all what the Weizmann Institute friends in Europe and in Switzerland are doing to help all the researchers in Weizmann. And they enabled the people in Weizmann to do the wonderful work. So thank you again for your dedication, for your engagement, and I really looking forward to see you again, not only on Zoom, but also in Israel in person. Thank you very much.
Thank you, Jacob, and really thank you for being such a long-standing friend of the Swiss Society. I think you participate at every one of our events, and also would like to extend my wishes to your wife, Dr. Osnat Kaidar. A lot of success in your future endeavors. We now have also the great pleasure to have Mr. Bob Drake with us. Bob Drake is the Vice Chairman of the Executive Board of the Weizmann Institute of Science, and he's also chairing the European Committee of the Weizmann Institute of Science. So that's the organization actually overseeing all the activities of the friends here in Europe. Bob, the virtual floor is yours.
Thank you very much, Eric. And I've got my feet squarely on the virtual floor. Let's hope it holds my weight. Thank you, Ambassador Kaidar. You have been with us every year for 5 years. What a great example of support, and best wishes for your future. And we certainly will meet in Israel at some stage. Dear friends, this year we are celebrating the 60th anniversary of the establishment of the European Committee for the Weizmann Institute of Science. We had hoped to celebrate this special milestone at the global gathering planned for last June, which, as you know, was postponed to June 2021. We hope that it will indeed happen, and we can celebrate in person soon. The creation of EQUIS, the European Committee of the Weizmann Institute of Science, in Zurich was historic. That's because since its birth, the Swiss Society of Friends has become a virtual pillar of support for the Weizmann Institute and a role model for the entire European family of the Institute. Friends throughout Switzerland and Europe have funded countless research projects across our beloved campus during 6 decades. When walking around the campus, which I hope all of us will be able to do again very soon, it is energizing and heartwarming to see the Swiss fingerprints. The tremendous impact is amazing. I would like to take this opportunity to thank you, our dear Swiss friends, and wish good health and many more years of celebrations around great science and the joy of supporting the Weizmann Institute. As you may know, in Hebrew, you wish long life to someone by saying bis 120, or until 120 years. So it's easy math to say, let's double it, and in another 60 years, we will be wishing EQUIS happy birthday 120. Thank you very much, and enjoy the rest of the evening.
Bob, thank you so much for your words, your dedication and contribution for chairing the European Committee, and also, you know, your very strong support of the Swiss Society. We really appreciate that. Now, I'm also honored to hand over the virtual floor to Professor Alon Ken. He is the president of the Weizmann Institute of Science. Alon just took over at the end of last year. You know, probably no one could anticipate what he had to expect when he took over. He thought he gets a well-run institute to take over, and then Corona crisis hit him, and now he had to do a lot of things ad hoc, which probably he would have not expected, but he's managing that extremely well. Alon, the floor is yours.
Thank you very much, Eric and Bob. Good evening, everyone. Great to see you all. It is my pleasure to greet you on the occasion of the Swiss Society annual Weizmann Lecture. Now it's 20th year, which is indeed a special milestone. As Eric already mentioned, as you know, next month I will be completing my first year as Weizmann president. And believe me when I say I had all sorts of scenarios about how my first year as president will turn out. And I can guarantee you that this wasn't one of them. So I have heard so much about this annual gathering, and I'm really excited to take part. It offers me also an opportunity to express my gratitude for the incredible devotion of our lay leadership and our many Swiss friends. The connection between the Swiss society and the Weizmann Institute goes way back over decades. Throughout this time, the Weizmann Institute has established essential research centers, institutions, and countless other projects and programs across all fields of research, thanks to the partnership of the Swiss society. I know that I and our entire Institute leadership can always count on the willingness of our friends in Switzerland to stand alongside the Institute and assist in fulfilling its mission of advancing basics research for the benefit of humanity. An example of this generous devotion is the establishment of the Swiss Society Institute for Cancer Prevention Research at the Morris Integrated Cancer Center. The Swiss Institute has become a vital pillar in the critical research around finding ways to prevent cancer before it begins. Today, it is clearer than ever that science saves lives. The world is looking to science and medicine for solution to the coronavirus pandemic. At Weizmann, more than 60 projects are underway focusing on developing faster and safer tests for detecting the virus, helping reveal its structure and genetics, and improving methods for contact tracing and tracking the spread. Our scientists have able to advance these projects, working day and night, each from their own angle and area of expertise, because of the environment of academic freedom in which they are working. Our supporters and friends enable this environment by kindly contributing to their research and supporting the Institute in its mission overall. In short, we could not do all that without you. While we are actively pursuing coronavirus research, we are simultaneously advancing our 3 flagship projects. And I would like to share with you, in a nutshell, the essence of these projects. The first flagship project is the Artificial Intelligence Enterprise for Scientific Discovery, which is a project that will build on the Institute's prominence in mathematics and computer science. As you may know, one of the biggest challenges that science is facing now is the sheer amount of data that is being generated and making sense of all of it. We are swimming in data now in drug discovery and genetics and climate and environmental research and astrophysics and many, many others. Only sophisticated AI tools will enable us to get our arms around all that information, produce significant understanding, and offer the best possible application in a range of fields. We are very excited about one special new development that will be part of our flagship project in artificial intelligence. The new normalization agreement between Israel and the UAE has enabled us to create new scientific partnerships. In September, as you may heard, we signed a memorandum of understanding with the Mohammed bin Zayed University of Artificial Intelligence in Abu Dhabi, and we will soon travel to the UAE to discuss the detailed agreement. A second flagship project is the Institute for Brain and Neurosciences, an initiative that will bring about 40 groups on campus together. Not only neuroscientists, but also scientists whose work has major relevance for the field. They will come together in a modern building that will enable collaboration throughout its physical structure. We are going to literally build the future of neuroscience and work hard to make many brain and neural diseases an history. Last but not least, the Frontier of the Universe project. Now, we'll leverage our expertise in astrophysics and particle physics to study phenomena at the grandest scale of the universe, all the way to the tiniest scale of particles. A central feature of the Frontier project will be Ultrasat, which is a satellite with a tiny but mighty telescope on board. Ultrasat is designed to alert ground-based telescope to transient event in space, like supernovas and formation of black holes. So I'm confident that thanks to the strong bonds between the Institute and our European and Swiss friends, we will be able to achieve these goals and more. I hope to see you in person next year in Zurich for the annual Swiss Gala. Thank you very much for your support and friendship, and enjoy the rest of the evening program. Thank you very much.
Alon, thank you so much for attending today's event and for your warm words.
Thank you, Eric.
Well, we are unfortunately unable to meet in person this year, as we mentioned before, but we wouldn't be the Weizmann Institute if you would just take that as a challenge which we cannot meet some new opportunities. And the opportunity we have today is that we will get a virtual visit to the lab of Professor Selenko and to gain an insight into his daily work. As mentioned, we have planned time for questions. So you can write questions into this link on the Zoom, the chat function. And Miss Evelyn Thalmann, Vice Chair of the Swiss Society, will then present your questions. Will also be the possibility to then unmute and to ask the questions directly, but maybe it's easier that if you just type it into this function. But we leave it up to you, whatever you prefer. Now, I would like to introduce Professor Philipp Selenko, the speaker of tonight. Professor Philipp Selenko is a native of Austria. He received his Master in Science in Chemistry and Biochemistry from the University of Vienna and completed his PhD at the European Molecular Biology Laboratory at the University of Heidelberg in the year 2002. He was a postdoc fellow at Harvard Medical School. Before joining the Department of Structural Biology at the Weizmann Institute, he headed the InCell NMR group at the Leibniz Institute of Molecular Pharmacology in Berlin. Philipp uses nuclear magnetic resonances, or NMR, spectroscopy to determine the three-dimensional structure of proteins directly in live cells. Similar to an atomic resolution microscope, he developed his methods to study the structural behavior of proteins involved in human neurodegenerative disorders, such as Alzheimer and Parkinson's disease. Specifically, he analyzes how these proteins change their shapes and structures and how this process is related to aging. These discoveries may ultimately enable the design of new drugs against Alzheimer and Parkinson's disease. His academic and professional honors include fellowships on many, and including from the European Molecular Biology Laboratory in 1998, European Molecular Biology Organization, EMBO, in 2003, Human Frontiers in Science Program Organization in 2004. In 2007, he was awarded the Max Kade Fellowship by the Austrian Academy of Science. He received a number of major grants, including the Emmy Noether Program grant by the Deutsche Forschungsgemeinschaft in 2007, and then the SAW grant of the Leibniz Society in 2010, and European Research Council Consolidator grant in 2014. Well, now enough with this long list. I mean, it's really impressive, but now let's have a visit to the lab of Professor Selenko.
My name is Phil Selenko. I'm a new faculty at the Weizmann Institute of Science. I joined the institute about 2 years ago after having run a group of scientists for more than 10 years in Berlin, Germany. And we decided to relocate to Israel and to the Weizmann in particular, especially because of the amazing opportunities that we saw for us here in terms of collaborative efforts with other groups at the Machon. My group is interested in amyloid proteins and human amyloid diseases, which mostly affect an aging population. I'm sure most of you have heard about Alzheimer's and Parkinson's disease. Those are the most common and prevalent neurodegenerative disorders that affect a huge amount of people worldwide. What my group is trying to understand is how the main protein players in these diseases change their structure and their shape in certain types of neurons and begin to form amyloid fibrils that will eventually deposit in certain brain cells and lead to the disease phenotypes that manifest in those disorders. For this, we have developed a new type of microscope, which really allows us to zoom into the cell to the highest possible level of resolution, which is down to the atomic structure of those proteins. And we are following those proteins in real time, directly in cultured neuronal cells. And we ask how they change their structure in the course of developing this disease. So we're really trying to record a movie, if you wish, of how the amyloid cascade and the formation of amyloid fibrils in certain types of neuronal cells happens in real time. For this, we use a new type of microscope that we've developed over the past years, starting at Harvard Medical School when I was a postdoc, and then further developments in Germany. And now we are applying this technology here at the Weizmann Institute in collaboration with lots of other groups to study and learn about these disease processes. So the special thing about our microscope is that it doesn't work with optics, it doesn't use light, it has no lenses, it doesn't work like a conventional microscope, but it's really based on a completely different concept, and the concept is quantum mechanics. We essentially exploit quantum mechanical properties of nuclear spins that behave in a certain way in a very strong magnetic field. And if we add radio frequency pulses to those spins in a very strong magnetic field, we will make them flip and behave and dance around in a very particular way, which depends in essence on their three-dimensional structure in the proteins that we want to study. So using this type of concept, which is a radically new idea of using nuclear magnetic resonance spectroscopy for visualizing proteins directly in cells, we can essentially arrive at the atomic resolution level, so at the highest possible level of resolution we can achieve to visualize the three-dimensional structure of proteins directly in live cells. And another trick that we need to do in order to study one particular type of protein at a time against the backdrop of everything else inside a neuronal cell, containing all the biological components that obviously make up this particular cell, but we only want to study one of this component, which is the amyloid protein that we are interested in. And so in order to achieve this, we specifically enrich the amyloid protein with heavy isotopes. Those are non-radioactive, but those heavy isotopes make the protein dance in a very specific way in the microscope that we are using. And the microscope is tuned to specifically pick up the dance of those proteins and blank out, if you wish, the background of all the other proteins inside the cell. And this is how we can specifically follow how individual amyloid proteins engage in structural rearrangement and eventually grow into amyloid type of fibers. And the main reason for wanting to understand this process at the atomic resolution level is that we wish to understand which parts of the protein change their structure and change their shape first. What triggers this whole cascade? If we understand this process at the highest possible level of resolution, we will eventually be able to design new drugs, novel types of therapeutics that can specifically intervene with this kind of process at the very beginning and thereby prevent the amyloid cascade altogether. And with this, we hope to be able to contribute to the development of new drugs and new compounds that will at some day be used in the clinic to treat, reverse, and prevent the occurrence of those types of neurodegenerative disorders. And if one looks at the prevalence of those diseases in the world, one understands that this is a truly pressing problem. Overall, people are getting older. They want to maintain a high quality of life until later stages also of their life. And this is why fighting these disorders is of such a huge importance. And we really hope that our science will be able to contribute to finding new cures for diseases like Alzheimer's disease or Parkinson's disease. Doing our research at the Weizmann Institute of Science, we have an amazing group of colleagues that provide complementary techniques and expertise that constantly feed into our own research. We thrive on interactions with groups in physics, chemistry, as well as biology, which provide a setting and an environment that is not only very stimulating to do research, but also hugely beneficial for progressing our own experiments. Working with other departments and core service units here at the Machon allows us to integrate the technology that we are using with different types of methods and techniques, and thereby harness the combined advantages that different types of methods provide. Apart from international collaborations that we maintain and foster, and of course reach out and provide expertise to other groups in the kind of technology that we are currently developing and establishing here at the Weizmann Institute, we also enjoy a great deal of support and interactive collaborations with other groups at the Machon. And these interactions range from departments and groups in physics to chemistry to biology. And the degree of resources and the types of technologies, complementary technologies that we have available at the Institute is really unique, which propels our own science so much further than working in isolation. And I think this is one of the strongholds of the Weizmann Institute, that the level of expertise and technologies present on campus is unique within the world, and it is so much fun and it's such a great joy to collaborate with groups on campus and beyond.
Philippe, the floor is yours.
Okay. Well, thank you very much, first of all, to all the colleagues here in Israel and all the friends in Switzerland and abroad. It's a great honor to be presenting to you today. At the same time, we all suffer from the circumstances, and I do understand that we would all have preferred to meet in person and to interact much more closely, but unfortunately with the current situation, as we all know, this is not possible. So we dearly hope, and I think I speak for all of us, that in the future we will be able to meet again in person and sort of look back at 2020 and hopefully have forgotten most of the bad things that this year brought, and remember the good times and the good things that happened also during 2020. I have to be honest with you, it wasn't even easy to shoot this clip because it happened during the lockdown. As you know, we had a second lockdown, and there were times where the lockdown was very strict. We were not allowed to bring anybody on campus. We were not even allowed to, be in the lab. So bringing a camera team in already was, a big effort, and we had to actually shoot very, very early in the morning before anybody else of the few people that were allowed in the building arrived, so that we could do this. Clearly, we had hoped to provide you with much more visual footage about the kind of things that we do, but unfortunately, again, due to the circumstances, this was not possible. Nonetheless, I do understand that working on neurodegenerative disorders is something that is dear to the heart to many people, because very often when I speak about those diseases, there are always people in the audience that have either a family member or a friend that suffers from those diseases. So I would be very happy to answer questions both in English as well as in German, if that makes some of you makes you feel more comfortable. Nonetheless, before I do that, I also want to mention, because I'm looking at the names here on my screen, and of course I remember many of them, and I want to send my regards to both Manfred and Ingrid from the German chapter, and we've interacted very productively on the German tour. But there's one person in particular that I would like to point out because of purely scientific reasons, and that is Kurt Wüthrich. I see his name on my screen, and what you need to know is that Kurt Wüthrich was a professor at the ETH in Zurich, who essentially is the founding father of modern nuclear magnetic resonance spectroscopy. I think Kurt is currently a professor at the Scripps Institute in California. However, what you need to know is that for his developments in NMR spectroscopy, which clearly form the basis of whatever we do in the lab with the type of microscope that I was talking about. So the whole concept of this microscope, the basic physics of how this microscope works, was developed by Kurt Wüthrich, for which he got in 2002 the Nobel Prize in Chemistry. So for me, seeing Kurt's name here on the screen is an amazing honor, because again, Kurt essentially paved the way in NMR spectroscopy about everything that we do on a daily basis. So it's great seeing you here, and I'm truly honored also that you decided to participate in this meeting. And with this being said, ich möchte mich natürlich auch noch auf Deutsch anschließen und allen Freunden, vor allem des Schweizer Chapters, herzliche Grüße aus Israel schicken. Wie gesagt, die Lockdown-Situation, die ja jetzt in Europa, auch in den deutschsprachigen Ländern, Schweiz, Österreich, als auch Deutschland mehr oder weniger eingetreten ist, die hatten wir soeben beendet. Ich möchte mich noch einmal auch bei Ihnen fürs Dabeisein bedanken, und ich würde mich freuen, auch wenn Sie die Fragen in Deutsch stellen wollen, kann ich die Ihnen auch gerne in Deutsch beantworten. Vielleicht nimmt das ein bisschen die Scheu vor diesem digitalen Format. Wie gesagt, sowohl Deutsch, Englisch, Italienisch wird ein bisschen schwer, Hebräisch auch noch nicht so ganz, aber ich würde mich jedenfalls freuen, möglichst viele von Ihren Fragen beantworten zu dürfen. Vielen Dank.
Now it's up to me.
Okay, so thank you, Philip, for your insights, and in particular the walks through your lab. Now I would like to hand over to Evelyn Thalmann, to run through the questions. He really got the number of questions, and let's start with that.
So hello to everybody. Do you hear me? Is it okay? Yes.
I do, yes.
Super, super. So, but first of all, Philip, I would like to ask you, why did you choose the Weizmann Institute of Science in Rehovot, Israel? I mean, Vienna is also a very nice place to be there. And there are also many other places in Europe or in the States. So tell us, why are you in Israel?
So for me, this was truly a professional decision. I looked at places on an international level where I would have liked to do science, simply because the environment is very stimulating, as I mentioned. And I think for science to progress well, you really need to have a lot of different expertises together. And coming across and looking at different places that offered such opportunities, the Weizmann Institute to me really stuck out as a unique opportunity, with the kind of ideas that we had to apply a method that is rooted in physics, but applied in a very biological kind of sense. We really needed to bridge certain disciplines from again, physics to chemistry to biology. And one place where this, to me, seemed immediately possible was the Weizmann Institute, because it has such a strong hold in so many of the core disciplines in the natural sciences. And so at the end of the day, the decision for me was very easy to make. I was convinced that we can do the best possible science here at the Weizmann Institute. And so when I presented my group in Germany with the decision that, that I will accept the offer to come here, of course, many people were surprised at first. It was not always an easy sell, so to speak. I had to convince many of my coworkers to, lose their fear of coming to Israel and doing great science here. But at the end of the day, I think everyone has understood that, that the way that this decision was made, again, purely based on professional and scientific reasons, was really the best decision that we as a group of scientists took at that particular moment in time.
Mm-hmm. Thank you so much. So there's another question I would like to raise. I mean, we are here in Switzerland. Are there already any cooperation with research institutes in Switzerland? Do you work with them? ETH or?
We do indeed have lots of very active contacts to research groups in Switzerland. Again, because one of the nuclear magnetic resonance spectroscopy is very closely related to Swiss science. I mentioned Kurt Wüthrich, who got a Nobel Prize. There's actually a second Nobel Prize on nuclear magnetic resonance that was awarded to Richard Ernst, who is another Swiss researcher. So the ETH and the NMR department at the ETH has an amazing reputation in nuclear magnetic resonance spectroscopy, and we are still collaborating with actually two groups there. We have connections to people at the University of Zurich. We have connections to people at the EPFL in Lausanne. So on a professional level, yes, our ties to Switzerland are particularly strong. And you may be surprised to hear that even on a personal level, I know Switzerland very well because my sister has lived in Zurich until the last year before she moved with her family to the United States. And so I've been to Zurich many, many times, not only for scientific reasons, but also for family reasons. And I truly connect to the place. And, I love the people, I love the country, and the science there is obviously absolutely amazing as well.
Thank you. Thank you very much. So I go into all the chat questions I got, and so I will read it now. So there is from Raul Bloch a question, and he said, hello, Professor Selenko, you applied your innovative microscope to analyze amyloid proteins. Are there other fields in chemistry or biology where you can apply this technology, maybe in the context of other teams?
Yes, absolutely. I think this is a wonderful question, and also to be scientifically accurate, I need to explain that we chose the term microscope as an analogy that is easily accessible for most people in the audience. Strictly speaking, this is not a microscope. Strictly speaking, this is a spectroscopy technique. But I think a microscope is easier to understand for what we do, and that is really look at very, very, very tiny objects. And protein NMR, so the types of structures that we are studying with this type of microscope, can of course extend also to other disciplines. If we think about many of the catalysts in nature, they are actually based on proteins as well. And many of the enzymes that, allow and give us our metabolism as we know it, that allow us to burn, sugars to create energy, for instance. Most of those biological catalysts are proteins as well. And indeed, they can be studied with this particular machine equally well, both outside of the cell as well as inside of the cell. So the beauty about this machine is that it's not necessarily and entirely and only made for looking at things inside of the cell. You can also look at isolated, purified components outside of the cell. And again, those components may have functions in different areas of natural sciences. And so the field of applicability is actually very broad.
Mm-hmm. So I do have another question, that is, there is a position that amyloid is not a cause for Alzheimer, but a protection mechanism that does not need to be attacked by enhanced or dealt with.
Yes, that's also a very good point, and that relates to the toxicity of those amyloid fibers that eventually grow into those very large structures that deposit in the brain cells and lead to the clinical problems and symptoms that we see in those patients. And one of the theory says, and there's very good evidence for this, is that once those structures have grown above a certain size, that they are no longer toxic to the cell, but it's really a way for the cell to deal with those humongous aggregates. And that what actually is toxic to the cell are much earlier types of oligomers that are forming on the way of becoming one of those very large structures. And again, there's very good evidence that the final structure, the mature amyloid itself, is not what's causing the problem in these diseases. And this is exactly one of the points that our science wants to address and wants to target. So we are really interested in the very early events of this cascade. In other words, in those initial structural rearrangements that lead for 2 or 3 or 4 protein molecules to come together in a very specific manner so that they form the seed of what is going to eventually grow into this amyloid structure. And I think once we understand those very, very first steps of the cascade, even if the endpoint of the cascade is not cytotoxic, but if we have a good understanding of the first steps, if we can interfere with the first steps and can block the cascade altogether, then essentially we will also prevent the accumulation of oligomeric species that may be the cytotoxic species, and prevent at the same time also the formation of the aggregates. So in essence, whether the final structures are really the cytotoxic species or not is going to become irrelevant, because once we block the whole cascade at the very early time point, we don't really need to worry about anymore what's going to happen later on. So that is also one of the idea of using such a high resolution instrument to do the science that we do.
It's so impressive, Philip, really very much impressive what you said. And I come to the next question. This is from Noah Lawrence Shapiro. And it says, I'm interested in your opinion on using stable isotope labeling kinetics as a biomarker for amyloid beta and tau, question mark. Do you think this will be common practice in the clinic soon?
So I think I have to probably define this question a little in a little bit of a more detail. What Noah probably means is that when we go to the hospital and we get a PET scan or we get an MRI scan, I mean, this technology, is the MRI technology especially, is very, very similar in terms of the physics that we are using to the kind of instrument that we do. Most of us who've been in an MRI magnet will know that what we get out in the end is a beautiful scan of our head or of our torso, where we get pretty good resolution in what goes on inside of our body. For this MRI technology, we don't actually need any contrast agents in most cases. The contrast of the images is given by the different types of water behaviors that we have in our body. So our kind of microscope works a little different, but exploits the same type of technology. And the fact that we use stable isotopes means that also those isotopes do not actually decompose into like radioactive isotopes would. So in terms of doing kinetic type of experiment, if I understand the question right, this actually doesn't work with those isotopes because over time, they don't change their chemical composition.
Mm-hmm. Okay, so that gives us another insight. So the next question is, just that I'm going to take everything what I have into. I'm wondering, so there are so many questions, so I have to click it first. So I'm wondering what is known on the effect of the strong magnetic fields on the RF pulses on the cytological processes. It is known that cells respond to magnetic field. That's Reto who raised that question.
Based on everything we know so far, strong magnetic fields do not exert measurable effects on cellular metabolism of individual cells up to humans. Migrating birds, for instance, can read very strong magnetic fields very differently than humans, but based on current knowledge, humans are not affected by strong magnetic fields. And the same is true by the radio frequency pulses that we use. Those radio frequency pulses, in terms of their energy input as well as output, are actually really weak. And there are no true measurable effects on you know, the biology that goes on inside the cell. The only thing that they do, again, depending on the type of sequence of pulses that one uses in those experiments, is they can induce slight heat effects, which are very local, but this is only over a few degrees. And the probe which is inside the instrument actually actively controls the temperature in the sample and compensates for temperature fluctuations, but the effects are really and truly minute. So by everything we know today, neither strong magnetic fields nor radio frequency pulses disturb the cellular environment in any measurable manner.
Okay. So, and there's a question from Leo Wiprinski. Do you see a connection between lifestyle and toxic structures on microscope level? Meaning, do you see a correlation of lifestyle and effect on cell level that is significant? If yes, are there striking examples?
So lifestyle on a cellular level, you would probably translate into metabolic changes that even a cell can experience. And the answer to this question is yes, indeed. So when we look at cellular aging, what that means on a molecular level is essentially accumulation of toxic species that naturally accumulate in every cell that has to produce energy, that has to metabolize nutrients. So there are always some toxic byproducts. As long as the cell is young, the cell can deal with those toxic byproducts in a very efficient manner. But the older a cell gets, the more cell divisions it has experienced in his life, even in a Petri dish, the less capable the cell gets in repairing those toxic byproducts that just accumulates, and which means that their levels get progressively larger, even in an aging cell, as it does in an aging person. So there are lots of similarities between metabolic changes on a cellular level, as well as metabolic changes that we experience on the level of an organism. And we can tune and we can induce sometimes those changes at will. If we want to mimic aging, for instance, on a cellular level, we know that we have to treat and expose the cells in a certain manner so that we can recapitulate metabolic conditions that also an aging or an older cell would experience. And by doing this, we can recapitulate types of cellular aging even in a Petri dish or inside our microscope to induce structural rearrangements that those amyloid proteins may experience.
Mm-hmm. So in your research vision, do you wish to try and reverse Alzheimer's back, or to be able to stop further deterioration?
So ideally, we would like to be able to do both. So we would like to understand the processes that lead to those disease processes in the first place. And if we can somehow intervene with them, then we can probably prevent the occurrence of those diseases altogether. At the same time, we would like to be able to revert the disease if it has probably already started inside the cell. And if we have accumulated seed structures which can potentially develop further into mature amyloid fibers, if we can find compounds or drugs that can actively, either prevent the further growth into mature amyloid fiber or even revert what has already formed inside cells. I mean, that would be a fantastic way to counteract the disease, both in a preventive as well as a manner that can serve as a cure. Mm-hmm. Even if we just help the cell with its own mechanisms to better deal with the amounts of toxic structures that that accumulate during aging. So maybe we can boost some of the endogenous machineries to detoxify the cell, to better clear toxic substances, and to be able by themselves to deal with toxic aggregates of those proteins that may accumulate during the onset of such a disease process.
It's a long way to go, isn't it? So there are other questions from a lady called Eva. And how do you enrich specifically amyloid proteins with heavy isotopes? Yes,
that is a very good question. We do this outside of cells. In other words, we produce those isotope labeled amyloid proteins outside of the cell, and then we introduce them selectively into the cell. And thereby we guarantee that the only thing which is isotope labeled inside the cell is really the component that we are interested in. So it's a two-step process. In order for this microscope to selectively pick up what we want to see, we purify those components, we isotope label them, and we put them back into the cell.
Okay. And what is the function of amyloid formation? It's also Eva that asked that question.
It may be a way of the cell to deal with toxic aggregates. Amyloid cascade might be a very reversible process throughout our entire life. And then there seems to be a tipping point where probably the amyloid cascade can no longer be reversed or prevented. And then it grows into those large aggregates that eventually deposit in the brain. It is a good possibility that abnormal protein aggregation happens all the time as a side product of metabolism inside the cell. Mm-hmm. But it seems that cells are, as long as they're young and healthy, they're perfectly capable of dealing with those events. It's only when the cell gets older and loses its capacity to repair itself and to scavenge those toxic aggregates that we are developing those types of problems. And this is why most of those diseases are really age-related and primarily affect an aging population.
Mm-hmm. And a very, let's say, common question, everybody wants to know that, is do we need drugs, or can humans stay healthy with a lifestyle where they move enough and stay mentally active so that we are our own doctor?
I think a healthy lifestyle definitely, helps to age in a good quality manner. That is commonly true for many, many diseases, and also true for neurodegenerative disorders. However, even a healthy lifestyle cannot guarantee that we will not develop those kinds of diseases. So in general, I would definitely proclaim a healthy lifestyle, enough exercise, fresh fruit, fresh vegetables, everything which keeps those toxic metabolic byproducts from generally accumulating in our cells. I think this is really the best anti-aging therapy that there is. However, again, even the healthiest lifestyle does not guarantee that one will not be affected by those disorders and diseases.
So coming back to the Institute, and I would say this is the last question I can raise, is do you collaborate with any of the labs connected to Alzheimer on the campus? If yes, with whom and in what way? In which way?
There is a fantastic group of people working on those types of disorders, and from all different sorts of angles. And just to mention one lab, Michael Schwartz, for instance, she is interested in how the immune cells of the brain respond to those toxic aggregates forming in other types of cells. So those are neighboring cells that suddenly begin to feel that an amyloid is growing in a cell, and the cell is sending out stress signals. It sort of asks for help to deal with this kind of insult. And the immune cells of the brain, they begin a process which is called neuroinflammation. And neuroinflammation is there to sort of seek help to detoxify the particular cell that is affected by this process. And Michael has found some amazing connections between the activation of neuroinflammation and the onset of aggregation and amyloid formation. So with her group, for instance, we constantly exchange ideas of how we can probably even create a situation in our microscope, so to speak, that we have two types of cells there. Not only the neurons that contain the amyloid proteins, which is on the way of aggregation, but probably also the sort of the immune cells of the brain next by, and see how their presence and their active signaling to counteract the aggregation process, how this affects, again, the three-dimensional structural rearrangements that those proteins undergo. So there is multiple interactions with different groups on campus, which is exactly, as I said also in the clip, what makes our science so enjoyable at the Weizmann.
Yeah, super. Now the really last question I just got from Ralph Friedlander. Could your innovative microscope be used also to visualize prion diseases like Creutzfeldt-Jakob disease or transmissible spine-form? Oh, now it's
encephalitis. Yes.
Encephalitis. Yes.
Yes.
Right.
This, for instance, is a process that Professor Wüttrich, who I mentioned before, has studied to great detail with reconstituted purified components in vitro, so outside of cells. So this microscope is perfectly well suited to study also these types of aggregation processes. We haven't looked at these processes yet inside of the cell. So doing stuff inside of the cell is always a little more challenging than doing it outside of the cell, where one can control everything. But to answer your question, yes, absolutely. NMR spectroscopy is perfectly suited to study these types of proteins involved in all of those types of diseases.
Danke vielmals, Philipp.
Gern geschehen, gern geschehen.
Danke.
Yeah, thank you for Evelyn for taking the questions. And I think thank you for everyone participating. I think it was very lively, even more questions than sometimes when we meet. Thank you, Philip. I mean, you talked about disorders, but you had held a very orderly, speech and give orderly answers, and also movies. So thank you for that. So thank you, Professor Alan Chen, for participating. Thank you also to Bob Drake. And of course, last but not least, the ambassador of Israel and Switzerland, Mr. Jacob Kedar. We wish him the very best for his future. And also thank you, Evelyn, for co-chairing today's event. A special thank you goes to the people actually behind the scene making this event happening so smoothly. So thank you for the Equis team, particularly Hadar, Shira, and Shigeru. That's the team here in Switzerland and on campus, but also Ayelet Rice from the Resource Development Department on the Institute, Shabi Mualem from the Information System Division. So really these 5 individuals, I think, deserve a special applause. Let's do it like this. So, really thank you for participating at this event. We would be happy to hear any comments, whether you like the format, whether we can change something. I hope, nevertheless, that next year we can go back to the normal format, that we can meet again at the University of Zurich and see in person.